With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and micro-cap data place. Welcome to this special edition of the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered, FINRA-licensed broker-dealer and the source of the equity research available on Channel Check. This investor forum at the World Stem Cell Summit presentation features Longeveron, NASDAQ ticker symbol LGVN. Following a brief overview presentation from Longeveron CEO Jeff Green, Noble Senior Research Analyst Robert LeBoyer will moderate a Q&A session. If you registered for this event through channelcheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I'm pleased to present Jeff Green.
It's a pleasure and an honor to present the company today, and thank you to the Regenerative Medicine Foundation and to Wake Forest for hosting the World Stem Cell Summit, and to Noble Capital Markets for sponsoring the Investor Forum. Longeveron trades on the NASDAQ under the stock symbol LGVN and has a market value of approximately $124 million. We existed as a private company from 2014 to early 2021 and successfully IPO'd on February 12th of this year, so still a relatively new public company. At the end of the first quarter, we had approximately $24 million in cash, which gives us a runway to operate into the fourth quarter of 2022 based on current budget estimates. The company's lead clinical stage therapeutic candidate is a well-characterized living cell-based product derived from the bone marrow of young, healthy adult donors. And our development focus to date has been primarily on chronic aging-related diseases and conditions, such as dementia, frailty, and immunosenescence. As you can see on the slide, we have 4 clinical trial results anticipated this year, 2 in frailty, 1 shortly in hypoplastic left heart syndrome, and results from a sub-study in the metabolic syndrome. In addition, we anticipate initiating 3 phase 2 trials this year and an osteoarthritis treatment registry program in the Bahamas. So a very busy second half of 2021 for Longeveron. Specialized cells reside within various tissues of our body whose purpose is to repair, restore, and regenerate damaged tissues and organs. When we're young, we have an adequate supply of these tissues and they function as intended. Unfortunately, with age, we not only lose these cells, but the remaining ones are less effective. This is believed to be one of the primary reasons for age-associated increase in chronic disease. The benefit of modern medicine and better nutrition has resulted in an extension of lifespan of nearly 30 years compared to what it was 100 years ago. But that has not been paralleled by an extension in health span, or the period of time we live relatively free of chronic disease. In the US, the last decade of our life is lived in a state of poor health, often dependent upon others for care. Longeveron was founded on the premise that these specialized cells could be developed into an off-the-shelf biologic used to treat chronic aging-related disease, improve health span, and potentially to extend longevity. This well-known decline in MSCs in both humans and animals is associated with stem cell exhaustion, increased cellular senescence, mitochondrial dysfunction, and chronic inflammation, as well as increased susceptibility to infection, a state commonly referred to as inflammaging. Inflammaging, in turn, is associated, whether it's a cause or effect, it's debated, to most late-life chronic diseases and conditions. Aging, therefore, is the #1 risk factor for chronic disease. This is our very full Lomecel-B clinical pipeline. Our Alzheimer's disease program reported phase 1 data earlier this year and is now transitioning to phase 2. We have 2 phase 2 aging frailty trials in the US that were completed in early 2021, and we expect to report top-line data for both trials in Q3. We have a very interesting phase 2 Japanese aging frailty trial set to begin this year. This is an intriguing opportunity due to, 1, Japan's status as a super age society and the economic challenges that come with a single-payer healthcare system, coupled with the enthusiasm and regulatory framework that exists for cell and gene therapies in that country. We have an ongoing phase 1 study in acute respiratory distress syndrome due to either COVID-19 or influenza infection. And our one exception to our focus on aging We have a program for children born with hypoplastic left heart syndrome, which is a rare congenital heart defect that affects about 1,000 babies per year. In that phase 1 safety study, in that program, Lomecel-B is administered via direct injection into the heart. We have recently completed the phase 1 safety study and are preparing for the phase 2 randomized placebo-controlled study, which is funded by the National Heart, Lung, and Blood Institute. And finally, we sponsor an open label treatment registry trial in Nassau, Bahamas, under the authority of the Bahamian Ministry of Health, where eligible participants may travel to Nassau and receive Lomecel-B at their own expense at one of 2 clinics we have partnered with. Like the US, Lomecel-B is considered investigational and not approved for commercial sale. However, we are permitted to charge a fee to recoup our costs. This registry provides valuable and diverse safety and effect data that supplements the clinical data generated from our various trials. As previously noted, Lomecel-B is comprised of living cells that are isolated and culture expanded from the bone marrow of young, healthy adult donors. These donors are rigorously screened for viruses and pathogens prior to their donation. Donor screening adheres to US, EU, and Japanese testing standards. We isolate the MSCs from fresh tissue using a very specific cell surface markers, culture expand only this population of cells using a strictly controlled process to ensure a pure MSC product, and then freeze the cells in liquid nitrogen in pre-divided doses that can be delivered on demand in an outpatient setting through an infusion that lasts about 40 minutes. It can also be administered via local injection, for example, into joints or into the heart, which is how it is administered in the HLHS clinical trial. This type of product is referred to as an off-the-shelf product, and these cells can be administered to an unrelated recipient without triggering a harmful immune response because they are considered immunoevasive. Because of these product characteristics, we believe that we can manufacture Lomecel-B economically at scale while maintaining the critical quality attributes. It has become increasingly clear that the therapeutic efficacy mediated by exogenously supplied MSCs is secretory and primarily functional at sites of injury, disease, or inflammation, which is why many in the field, including us, are referring to these cells as medicinal signaling cells. So these 4 yellow boxes on this diagram indicate 4 potential mechanisms of action for Lomecel-B in the microenvironment. 1, the release of anti-inflammatory cytokines, which promote regenerative and repair responses. 2, through direct cell-to-cell interactions or through trophic signals with contacted cells. 3, the release of exosomes, which have cargo consisting of mRNA and other content. And 4, through nanotubule bridges, which can allow the exchange of mitochondria and other cellular contents between the cells. So I'd like to go into some detail about a few of our programs and share some of our data now. Starting with Alzheimer's disease. Alzheimer's disease, early and substantial involvement of neuroinflammation leads to neurodegeneration and neuronal death, which is a significant contributor to the pathogenesis of this deadly disease. It's been shown that in animal models, MSCs can cross the blood-brain barrier, reduce pro-inflammatory cytokines, and increase the anti-inflammatory cytokines, improve immune functioning, and promote both neurogenesis and endothelial function. Our hypothesis is that because of the pro-regenerative and anti-inflammatory properties of Lomecel-B, targeting neuroinflammation in the brain may prevent or reverse clinical progression. To that end, we first embarked upon a phase 1 study, which was funded in part by a competitive grant award from the Alzheimer's Association, and that trial concluded just late last year. This trial enrolled 33 PET scan confirmed mild to moderate AD patients who received a single infusion of either Lomecel-B 20 million cells, Lomecel-B 100 million cells, or placebo, and then monitored over 1 year. While the trial was intended primarily to evaluate the safety and tolerability of IV infusion, we evaluated various domains that included clinical endpoints, quality of life, biomarkers, and brain volumetry. So we successfully completed this trial and recently announced the results. First and foremost, the primary objective was satisfied in that we showed in this trial that Lomecel-B infusion was safe and well tolerated, but in addition, there was no evidence of swelling or edema, which is something that can occur and has been observed with the monoclonal antibodies. So we're optimistic that cell therapy will continue to show a very clean safety profile for the purpose of the risk versus the benefits. We saw some very interesting results with the lower dose 20 million cells, relating to the slowing of progression of cognitive decline as measured by the mini mental state exam. The graph on the left shows the decline in MMSE for placebo in the red line and low dose Lomecel-B in the green. Both groups started at about the same score, and the placebo-treated subjects showed immediate and significant decline, while the Lomecel-B group stayed fairly level through 6 months and then declined from there. The difference from placebo was significant at 3 and 9 months in this case. On the right is the ADAS-Cog11 results, and while not statistically significant, again, the low dose Lomecel-B had a slower decline when compared to placebo over the first 6 months, consistent with the MMSE. As you are all undoubtedly aware, Biogen's Alzheimer's drug was approved based on the reduction of plaques and the presumed slowing of decline in cognitive function. And then just speaking to the mechanistic effects of Lomecel-B, we observed improvements versus placebo in both provascular biomarkers of VEGF, IL-4, and IL-8, and anti-inflammatory cytokines, including SIL-2, IL-10, and IL-12. So we're encouraged and enthusiastic about these early results, and the program is now transitioning to a phase 2 trial, which we expect to start in the second half of 2021. Transitioning now to our aging frailty research program. Let me provide you with a little context first. Longeveron was actually founded on the premise of using cell therapy to ameliorate the geriatric condition of aging frailty. A phase 1/2 investigator-initiated clinical study conducted by the University of Miami Miller School of Medicine and published in the Journals of Gerontology in 2017 formed the basis for Longeveron's aging frailty research program. Frailty is a well-known and distinct biological condition that is characterized by a multi-system decline in physiologic reserve and function, chronic low-level inflammation, and poor clinical outcomes. Clinically, the frailty phenotype is most often defined as having 3 or more of the following domains: weakness, slowness, decreased activity, fatigue, and involuntary weight loss. Frail individuals do not recover well from a stressor such as a fractured hip, infection, or surgery. There is a loss of confidence in the ability to perform basic activities of daily living, like crossing the street or going to the market. As a result, they become dependent on others for care. Very, very importantly, frail patients are more susceptible to the complexity of the surgical process and are at a greater risk for complications after surgery, less likely to recover function postoperatively for things like cardiac surgery. The good news is that research has shown that early identification and intervention can improve or even reverse frailty and restore the individual to a more healthful state. So there are no products approved anywhere in the world to treat frailty, despite the fact that it affects up to 15% of the population over the age of 65. The cutout here is the American Geriatric Society guidelines for managing frailty, and it's really limited to exercise and nutritional supplementation. So frankly, there is not much in the toolbox for geriatricians when it comes to preventing or reversing frailty. Our frailty research program spans 3 clinical studies, 2 of which have recently completed, and a third will initiate in Japan in the second half of 2021. Our 2 US phase 2 frailty trials are both randomized placebo-controlled studies. The HERA trial on the left is designed to evaluate whether Lomecel-B can improve the immune response to flu vaccination in a population that typically does not respond well and are vulnerable to the flu each season. We measured antibody production early post-vaccination and over the course of a full year. In the second trial, we call it the phase 2B trial, we enrolled 149 subjects in 5 cohorts to evaluate the effect of Lomecel-B infusion on the signs and symptoms of frailty, with heavy emphasis on the physical function aspect of frailty. We anticipate reporting data in both of these studies in the third quarter of 2021. And the phase 2B study is, I think I should note, was funded by a sizable SBIR grant from the National Institute on Aging. And just for full disclosure, speaking of aging frailty as an indication, this is a new indication from a regulatory standpoint in both the US and Japan. So we'll need to review the data and discuss with FDA and the Japanese PMDA the pathway forward, which will include identifying and defining an approvable indication and the associated endpoints that would provide evidence of clinical benefit in the defined population. To Just a note, our Japan trial includes the nation's leading expert in frailty research, Dr. Hinori Arai, director of the NCGG in Japan, which is the equivalent of the National Institute on Aging in the US. In addition, we're working with one of the largest private hospital chains in the country, Juntendo University Hospital. A clinical trial notification, which is the equivalent of an IND, has been filed and accepted, and we expect to get rolling on that trial this year. Japan's Super Age Society and its cell and gene therapy regulatory framework make this an ideal scenario for Longeveron's anti-aging disease programs. Let's look at some clinical data in the Frailty program. From the HERA influenza vaccine trial, we also measured, in addition to immune response, physical function, and these are the interim pooled results of the 6-minute walk test. The red line is placebo, and the blue line is the pooled Lomecel-B subjects. So you can see that by 6 months, the Lomecel-B treated group, on average, could walk 75 meters further than the placebo group, which is a fairly large difference. Why is this important? Well, one, walking distance and walking speed are powerful predictors of mortality in frailty. And two, this is the primary efficacy endpoint of our larger phase 2b trial. So we'll hopefully see similar results when the final data comes out. We have preliminary evidence that suggests that Lomecel-B may augment influenza vaccine response in frail elderly subjects. Given what's happened over the past 18 months with COVID and the deaths of elderly, this has become quite an urgent and timely program for Longeveron. These are the results from the phase 1 open label HERA study, where all subjects received a 100 million cell infusion of Lomecel-B in advance of the high dose flu vaccine. So what's interesting here is that in the first 4 weeks, all subjects mounted an immune response based on antibody levels that were considered protective or exceeded the vaccine effectiveness threshold, which is indicated by the dotted line on the graph. Furthermore, that flu season, 2017-2018, the CDC estimated that the vaccine was just 18% effective in individuals 65 and over. So again, while we are optimistic, it's the placebo-controlled data that will be the most important, and that's expected in the third quarter. And then for now, we'll transition to the HLHS program. So this is a congenital heart defect that affects about 1,000 babies a year, and it is, without reconstructive surgery, 100% fatal. Even with reconstructive surgery, a large percentage of these babies will go on to need a heart transplant. So there are 3-staged operations is the current standard of care, and in our program, Lomecel-B is injected directly into the right ventricle during the 2nd stage, or known as the Glenn operation. And the hypothesis is, will Lomecel-B aid in the repair and regeneration of cardiac tissue and therefore improve ventricular function? So we've recently completed the phase 1 open label safety trial, in which we demonstrated that in 10 babies, the Lomecel-B injection was safe and well tolerated. There were no serious adverse events attributable to the injections and no major adverse cardiac events. So the exploratory efficacy data from that open label phase 1 trial is expected in the 2nd quarter of this year, so very shortly. And now we're transitioning to initiate the phase 2 trial in the 2nd half of 2021. And that's being led by Dr. Sanjay Kaushal, who is the division head of cardiovascular and thoracic surgery at the Lurie Children's Hospital in Chicago. And I should say that trial is being funded by the National Heart, Lung, and Blood Institute. So I just wanted to show you the University of Texas Health Science Center is the group coordinating this trial, and they've set up this phenomenal website dedicated to the study for parents of these children to learn more about the research and the study. So I'm just showing you a couple of screenshots from that website. So just a refresher on the near-term catalyst for Longeveron. So as I think you've gathered at this point, we've got a very active 2nd half of 2021. The blue circles on this slide represent the release of data. Shortly, the phase 1 HLHS trial and the 2 frailty trials in the 3rd quarter. And then the approximate initiation of 3 phase 2 trials, which are all anticipated for this year: HLHS, Alzheimer's, and our Japanese aging frailty program. Our intellectual property is comprised generally of 4 different areas that we focus on. So we have company-owned patent applications, which are generally geared towards methods of use. We have licensed in, issued, and pending patents on a subset of MSCs that are still in the preclinical phase. We exclusively license the manufacturing process from the University of Miami, and in cell manufacturing, the phrase is often the process is the product. So we have a proprietary process for the production of our Lomecel-B, and we do our own manufacturing in-house, and we do not outsource this manufacturing to a third party. And then, of course, we would take advantage of any statutory exclusivity that would be available to us through any approvals or FDA incentives. We have an excellent team of seasoned executives, manufacturing experts, and scientists. This group has, for the most part, largely been together for the last 5 years or so. So this consistency has, I think, led to some of the success that we've had, and we continue to add to this team and grow its experience. And very pleased with how the team has come together post IPO. So in summary, I'd just like to say that we have a very well-developed MSC product called Lomecel-B. We've treated over 360 subjects to date, and so we've got very well-characterized safety profile thus far. We focus on aging-related chronic diseases and life-threatening conditions. We have our own state-of-the-art GMP manufacturing facility for stringent manufacturing of our product. It's been very safe and well tolerated to date, and we have supportive early clinical efficacy data and a robust clinical pipeline. We operate a revenue-generating Bahamas treatment trial program, and as you've just seen, we have a number of significant value inflection milestones over the next one year or so, and a very proven management, scientific, and manufacturing team. So with that, I'd like to conclude the presentation, and I'll hand it over to Robert for Q&A. Thank you.
Jeff, thank you very much for that presentation. You have a lot of interesting data and some groundbreaking applications in some important diseases. You noted that Longeveron's current cash is anticipated to last into the fourth quarter of 2022. What would you say are the next top 3 value-creating events or catalysts that could occur between now and then?
Yeah, great question. Robert, as an analyst, you know that biotech and pharmaceutical companies invest heavily in R&D for the purpose of defining and understanding the safety and efficacy of their product in the target indications of need. So it's often the announcements or the reporting results in rigorously designed and conducted trials that will typically be the value-driving events, followed by the transition into later stage trials as this signals further de-risking of the program and therefore the security, presumably. So to that end, we have 2 phase 2 trials, both in frail elderly patients, reading out in Q3 2021. And one of those is looking at immune response following influenza vaccine, and the other is a larger NIH-funded study evaluating the effects of Lomecel-B infusion on the general signs and symptoms of frailty. So there was 2 events in the third quarter of this year. The next one will be the announcement shortly of the exploratory efficacy results of our phase 1 open label HLHS trial. And then I guess I'd say the next kind of rolled up value creating events or catalysts for this company would be successfully initiating 3 phase 2 trials this year, 1 in Japan for frailty, the next phase of Alzheimer's disease research, and then the phase 2 randomized placebo-controlled HLHS study.
And the entire field of cellular therapy and gene therapy are booming and expanding with lots of discoveries and breakthroughs. Several companies have progressed to phase 3 pivotal trials in a variety of indications. So how would an FDA approval of a cell therapy affect the company's prospects?
No, the field has really progressed, and that is evident in the amount of investment that's pouring into cell and gene therapy companies, both development and manufacturing. There are a handful of leaders that are transitioning to phase 2 or phase 3 and hopefully licensing applications for some very large and lucrative indications. And there is a tremendous activity in Europe and in Asia, with Japan leading the way there. So I think a rising tide is going to, lift all boats should any one of these late-stage companies achieve a full approval from a major regulatory jurisdiction. So Wall Street has its skeptics in the cell therapy field due to the lack of regulatory approval. So I understand that, but I think a success will tip the scales in favor of the regenerative field, and we will see significantly more interest in companies like Longeveron, who are perhaps a few years behind, but making progress and learning from the mistakes of others who may have gone before them.
Yes, clarifying the regulatory path and being able to follow that guidance would help you. So one of the other things was that you mentioned the approach for Alzheimer's disease. And could you compare what Lomecel-B is doing with some of the other therapies for Alzheimer's that are in development or in progress?
Yeah, so the field, so obviously we went over this in the presentation, but Longeveron is taking the approach where it's the neuroinflammation, whatever the cause of that neuroinflammation, it's the neuroinflammation that we're trying to address and suppress through the use of a cell therapy, of mesenchymal stem cell therapy. So as everyone in the world knows now, the FDA just approved Biogen's product for AD, which is a monoclonal antibody that is supposed to slow the cognitive decline. And the benefit has been the subject of significant controversy, as there is a risk of brain swelling with these types of agents. And so you've got the monoclonal antibodies sort of to clear brain plaques, which I guess Lilly, Eisai, Biogen. There are agents that are being tested to address amyloid. There are the neuroprotective approach like Biohaven and Athera and Cassava. And then I would say Longeveron is sort of the leader in the field in going after purely a neuroinflammation approach. A couple of other companies have tested cell therapy, including two groups out of Korea, one called Nature Cell, and they're actually approved in a single clinic in southern Japan to administer autologous injections that are derived from fat. It's a autologous fat cell program. And so their dosing regimens, their starting materials, their actual product are kind of all different in this case. And to my knowledge, they have not published any data. So I think that's what differentiates Longeveron in the field. And I think the Alzheimer's Association really thought the approach to neuroinflammation was a novel one, because out of the groups that applied for the $4 million Part the Cloud on Neuroinflammation grant, it was Longeveron that received the award, and which allowed us to conduct that phase 1 trial.
Yes, I think that's an important point, that you are looking at the inflammatory aspect rather than the end-stage lesion of amyloid plaques. Fragility and aging are not currently recognized as approvable indications by the FDA. So how would you plan to navigate the regulatory pathway? And what kind of primary endpoints do you foresee in your phase 2B trial?
Yeah, so the future of that program obviously lies in the strength of the data and the evidence and the risk-benefit profile. Specifically, can Lomecel-B positively impact key functional endpoints that are predictive of clinical benefit? So endpoints such as walking distance and walking speed and grip strength and balance. So ultimately, Robert, we're trying to develop a therapeutic that reduces the poor clinical outcomes such as falls and fractures and hospitalizations, and to preserve independence and quality of life. So in the phase 2B trial, we selected the 6-minute walk test as the primary endpoint, which has been a primary endpoint in many products previously approved by FDA. The 6-minute walk test engages the musculoskeletal system, the cardiopulmonary system, and the neurologic system, and is a good overall global assessment of function. But that said, we recognize that improvement in 6-minute walk test will need to translate into improved clinical outcomes. So it is a surrogate or intermediate endpoint. And once we have the data in hand, we intend to have these discussions with the FDA on the proposed pathway forward. This is a dysregulation of normal processes. This is extreme aging that's caused by, a number of underlying issues going on in the body. And we measure an extensive amount of biomarkers in our trials, trying to identify which biomarkers may correlate with the changes in physical function, so that we can kind of put together a full story, and that full story translates into either improved quality of life, improved functioning, or overall survival for these patients.
Okay, that sounds very good. And In other Alzheimer's and neurological deteriorating conditions, I've seen activities of daily living used as endpoints and quality of life. So I think that would be an important point. And as you mentioned, the inability to carry out these activities of daily life leads to custodial care and then institutionalization, so that the value of slowing down this deterioration is not only quantifiable in terms of emergency room visits and morbidities, but also in the cost of care. So I think you have many endpoints that could be used. And then in your immunosenescence trial for your HERA fragility trial, you've shown an improved immune response with Lomecel-B infusion over placebo following flu vaccine. Would you consider collaborating with both flu and COVID-19 makers, or what would you imagine the next steps to be in the program?
Yeah, so I guess the short answer to that question is yes. The placebo-controlled phase 2 trial, where we gave a sham infusion in advance of the high-dose vaccine and compared that to a Lomecel-B infusion in advance of the vaccine, that data are coming out in the third quarter. So, look, if we can show a robust immune response compared to the placebo group in a group of individuals who typically, their immune system, their aging immune system is not mounting the appropriate response, and it continues to leave them vulnerable and susceptible to infection. If we can show an improvement in that group of people, and it's statistically significant against placebo, I would want, obviously, to collaborate or have conversations with, the COVID, the influenza, the pneumococcal vaccine makers out there to see if we can collaborate on a program that would, combine these and see if we can improve the efficacy of those products.
How is Lomecel-B differentiated compared with other bone marrow derived MSCs in development?
It's often sort of a closely held secret about the process that each of the manufacturers undertakes to produce their products. So, we also keep that as a proprietary program. It's something that we licensed exclusively from the University of Miami and have spent years optimizing. And so we have our own set of characteristics and attributes and surface cell markers that we adhere to create Lomecel-B. And of course, we're optimizing the yield and the production so that we continue to go towards scale. And then in parallel, for all these indications, as you well know, you can't really move forward without potency assays. So all of these kind of combine to create an overall proprietary product. And I think each of the cell therapy manufacturers would kind of answer similarly that they have their own way of making it. They try to keep that a secret to the extent possible. And some, but very few have actual patent protection around the production process. So it is kind of a proprietary scenario.
And in the pediatric congenital heart defect program, it veers away from the company's focus on chronic aging related diseases. What's the plan for that program?
It's such a huge unmet need. It's really something that we're passionate about at Longeveron. We're really proud of the fact that the phase 2 program was fully funded by the NIH. The centers that are involved, the University of Texas as a coordinating center. I think as a company, our goal would be to get that approved so that we can get it in the hands of the surgeons and these children and families that need it most. So, we're going to continue moving forward with this program. We're going to apply for any and all appropriate regulatory categories that we can: orphan drug disease, breakthrough designation, those kinds of things that we may be eligible for. And all of that would be for the purpose of moving this as fast as possible. So, the trial should get started very, very quickly, and the centers are ready to go. And I showed you copies of the website, so we hope to enroll that expediently so that we can get the data and talk to the FDA about the next step needed in advance of a full approval.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and microcap companies listed.